Good afternoon. Welcome. Good afternoon. Good afternoon. Welcome everyone to our BME seminar series hosted by the Department of Biomedical Engineering at Columbia. I am Stephen Zhang and a professor in pathology, cell biology, ophthalmology, and the stem cell initiative. So we offer this lecture at a hybrid format. So there's about 100 people currently on Zoom. And our visiting professor would speak about 50 minutes or so and leave 10, 15 minutes for questions at the end. For the virtual audience throughout the presentation, please use the Q&A feature at the bottom of the screen, not the chat. Remember to use the Q&A to submit your questions. We'll try to answer them as many as possible during the Q&A session at the end. For the live audience, you have a chance to ask a question during Q&A, and then maybe either Dr. Jacobs or Dr. Gersbach will repeat the questions from the live audience so that the Zoom audience can also hear. Today, we're really honored to have Charlie Gersbach as a visiting professor. Charlie is the John Steuben Distinguished Professor of Biomedical Engineering at Duke. He runs the Biomedical Engineering PhD training grant at Duke, and also the director of the Duke Center of Advanced Genomic Technologies, as well as the director of the Duke Center for Molecular and Tissue Engineering. As a principal investigator, the director of the center, Dr. Gersbach has more than $10 million of NIH grant per year. So he came through the academic ranks, started his career in Georgia Tech, and then went to Scripps, and then since went to Duke. You may know that 2012 was the CRISPR version of genome engineering described. In 2013, Charlie described an improved version called epigenome editing. That probably will be the topic today. Not only has then pioneered genome engineering and epigenome engineering, Charlie also has treated numerous disorders, including successfully mouse models for Duchenne muscular dystrophy. So without taking too much of Charlie's time, I just want to, if I keep telling you about what Charlie's accomplishment, will take the next hour. But that's just on the highlight. For his work, he's awarded the NIH Director New Innovator Award, the National Science Foundation Career Award, the Outstanding New Investigator Award from the American Society for Gene and Cell Therapy, the Allen Distinguished Investigator Award, and introduction to the Fellow of the American Institute of Medical and Biomedical Engineering. So it's indeed a great honor and pleasure for us to have Charlie to spend the afternoon with us.
Well, thanks so much for the really nice, kind introduction, and also for the opportunity to come and talk to you all in person today. It's nice to do this for a change, and as well as teaching the stem cell class yesterday. So also nice to come and visit Columbia for the first time and meet with all of the faculty that are doing such amazing things in genome editing and genome engineering. So it's been a real pleasure. So today, there's a couple of different topics I was thinking about, but the main focus of the presentation will be on some of the work that we've done applying genome editing to muscular dystrophy, and I'm required to first show some of these disclosures. The one relevant for the topic today around gene editing for Duchenne has really been a partnership with Sarepta Therapeutics over the last couple of years, and we're appreciative of their support. But before I get into that, I maybe wanted to kind of give you the big picture scope of our research program for a second, which is, I would say that the larger goal is to attempt to turn genomic sciences into a field of genome engineering. And so what does that actually mean? Well, the idea is that genomics up until now has largely been a scientific discipline. We want to turn it into an engineering discipline. And so what does that mean? Well, if we think about other engineering disciplines, like computer engineering, for example, it's very well understood what all of the different parts of a circuit do. And you can go in and you can change parts out. You can predict what's going to happen when you put new parts together. And it's a very predictable system. That's really not the case until recently for genomics, right? Not only were the tools not available to go in and exchange pieces or add new pieces or replace pieces in a very specific way, but it was very poorly understood what would happen when you did so, right? And so the overall goal is to develop the tools to be able to rewire a genome at will, just like you would rewire a circuit board, but then also be able to apply those to answer fundamental questions in science as well as treat disease. And I'll make a quick plug that I think Duke has been a really fantastic place to be able to do that. Cam Leong, one of your faculty members, I owe him an incredible amount of appreciation for actually giving me the opportunity to go and start my faculty position at Duke about 13 years ago, where we've been working in this area. And it's been a really incredible environment at the intersection of sciences, engineering, and medicine, very close-knit on this centralized campus with a lot of major efforts and centers listed across the bottom here that Dr. Zhang just introduced. And so if any of you ever are interested or have the opportunity to visit Duke, it's, I think, a really unique place and kind of how tight-knit and interwoven the sciences and engineering and medicine are, in particular in the area around genome sciences and engineering. But to quickly summarize the kind of focus areas of my individual lab, there's kind of three different areas that we've been developing and applying these tools over the last 10 years or so. And that includes developing gene editing approaches and applying them to gene therapy, building off the DNA targeting capabilities of genome editing tools to think about programming other functions into the genome, like manipulating genome structure, epigenetics, and gene regulation, and then taking those technologies and applying them to programming cell fate specification and cell differentiation. And so when I thought about some of the things that I might discuss with you today, actually yesterday, I lectured in the stem cell course and really covered these latter two topics and some of our most recent work in those areas. And so for today, I thought for this audience that maybe I would give an update as to some of our progress in this first area of applying genome editing to correct genetic diseases. And just to make sure that we're, as quick background, that we're all on the same page, what's unique about gene editing to treating genetic disease is the ability to make precise changes to sequences in the human genome. Genetic engineering, gene therapy, gene delivery, these technologies have been around for decades. We've got a couple of approved products that are gene therapies now, and dozens of others in clinical trials. But for the most part, those approaches are just adding extra genetic material to cells and not actually correcting the underlying causative mutation. And so with the advent of new transformative gene editing technologies like CRISPR-Cas9, our goal was to go in and actually fix those mutations rather than adding extra copies of genes to the genome. As I think maybe was mentioned in the introduction, I kind of got involved in the gene editing field around 2006 when I moved into my postdoctoral training, which was many years before the CRISPR-Cas9 system had been adapted for genome editing. And so we started using earlier tools, things like zinc finger nucleases and TALENs you may be familiar with, that were somewhat difficult to work with and not always successful, but everything changed with the advent of the CRISPR-Cas9 system in 2012. And I think that probably many of you are familiar with this, so I'm not going to spend time kind of going over the background. But the way this system works is the Cas9 enzyme, represented in this cartoon as this large blue blob, pairs up with a small, about 100 nucleotide guide RNA that has a 20 nucleotide targeting sequence anywhere in the genome. And then based on that targeting sequence, it'll take Cas9 to a specific site. Once it forms that complex, Cas9 will cut the DNA, which then facilitates gene editing and changes to DNA sequence. I'll come back to that concept in a second. So when we started the lab at Duke in 2009, we wanted to not only develop new gene editing tools, but we were really enthusiastic to apply them to specific diseases to see if we could really start to develop things that would look like therapeutics. And we had to think carefully about, what disease area would we move into? And we wanted something that was difficult enough that it would give us a lot of work to do, some job security over the next 10 years, and overcome some significant challenges, but also that wasn't going to be impossible, something that we could see a light at the end of the tunnel of how gene editing could be applied to this disease. And the one that we landed on to put a lot of our attention on over the last 15 years or so was Duchenne muscular dystrophy. And there's a number of reasons why we thought that this disease would be particularly amenable and interesting to apply gene editing, and I'll get into those reasons in a second. But to quickly describe what this disease is, it's actually it is the most common inherited genetic fatal disease. It affects about 1 in 5,000 boys. The disease is caused by a mutation to the dystrophin gene. Dystrophin protein actually serves as basically the mechanical linker or shock absorber in skeletal and cardiac muscle. Shown here is this kind of rod-like protein. It links at the N terminus with the actin cytoskeleton, and at the C terminus at the cell membrane in this dystroglycoprotein complex, and basically holds the muscle cell together as the muscle contracts. Now, what happens in the case of Duchenne is that this linker is broken because of a genetic mutation, often in the middle of the gene. And as I'll show in a couple of slides, it's typically actually a large deletion of 1 or more exons that then shifts the protein out of frame, and so that linker is no longer present. The linker is no longer present, then after repeated rounds of muscle contraction, there's damage to the cell and the cell membrane, and after many rounds of regeneration, ultimately leads to muscle weakening, dystrophic muscle, depletion of the satellite cell progenitor pool. And these patients are usually in a wheelchair around age 10 or certainly before their teens. Shortly later, will end up on requiring a ventilator and often suffer from premature fatality in their 20s or 30s. So there are a number of reasons why we thought gene editing would be a particularly interesting approach to treat Duchenne. The first is the clinical need, which I just described. And so these patients up until now really have no other disease modifying therapies available to them. There's a number of other things that make this type of neuromuscular disease particularly interesting for gene editing. And one is the idea of the potential to edit in the progenitor cells of the muscle. So we were thinking at the beginning, not only could you potentially edit the muscle tissue and the terminally differentiated muscle fibers, but if you could also edit and genetically correct the stem cells of skeletal muscle, the progenitor cells, then they might continue to self-renew and be a continuous source of new cells that could contribute to gene correction in that muscle tissue. We'll come back to that in a little bit. But the idea being that if you could correct those progenitor cells and they continue to self-renew, that would lead to long-term dystrophin restoration. Another interesting feature of skeletal muscle is that these cells are highly multinucleated. So every muscle fiber, a single muscle fiber cell, contains hundreds of nuclei. And so one of our major concerns, and I get a concern in the field for in vivo genome editing and gene correction, is always going to be about efficiency. How efficient can you actually do that gene correction? But in this case, because every cell has hundreds of nuclei, we hypothesize that we may be able to just correct a few nuclei, and those could produce enough dystrophin protein to compensate for their uncorrected neighbors and create a dystrophin positive fiber. We were also, and so low levels of gene editing might be sufficient. Another interesting feature of the dystrophin mutations are that the gene mutation can actually be corrected by removing DNA from the genome rather than having to replace or fix it. And I'll come back to that concept in a second, but we know that that's a more efficient way to do gene editing in vivo, because basically removing things is always going to be more efficient than adding things or replacing things. Additionally, it turns out that the mutations are not evenly distributed across the gene. There's actually hotspot regions. One of the challenges of applying gene editing to genetic disorders is that even if a bunch of patients with the same disease all have mutations to that gene, If the mutations are all over in different places of the gene, then you're going to need a different gene editing composition to treat their disease. And that's going to be very difficult to move forward through clinical trials and through regulatory approval. So if you can find one composition and one editing approach to get to a large number of patients with diverse mutations, that could be very advantageous and desirable. And it turns out in the case of Duchenne, there are hotspot regions of the dystrophin gene that can be targeted and thus get to large numbers of patients. And then at the end, I'll talk about moving past this idea of removing parts of the gene to restore its function and talk about restoring a full-length dystrophin protein. Okay, so I want to explain this idea of correctable by non-homologous end joining. And so to explain what that means, I want to talk about the different ways that gene editing can be used. And so if you want to add or exchange something to the genome, the standard conventional method has been to use the gene editing nuclease, in this case CRISPR-Cas9, to cut the DNA, which then stimulates homologous recombination. And if you provide this extra template, then it'll get inserted into the genome. And that could potentially be used for gene correction. The problem is that this is a relatively inefficient pathway, and it's actually almost completely shut down in post-mitotic cells, which would include the skeletal myocytes and the cardiomyocytes that we would want to treat in this disease. So we were concerned that this was not going to be a viable option for gene editing in vivo in this type of disorder. So that means we can't go in and correct the mutations, but what about these other options? So if you can't use homologous recombination, the other DNA repair pathway that is very highly active in these cells is non-homologous end joining, which basically means grabbing the two ends of the DNA and ligating them back together. But what we're going to be particularly focused on in this application for Duchenne is actually making two breaks in the genome in neighboring regions. And if the middle piece here floats away and those two distal ends get ligated together, then you can make precise targeted deletions to the genome. Now, why would we want to do that in Duchenne? Well, what I'm going to show you is the ability to use exon excision in order to restore a functional dystrophin protein. And the reason for that is, as I mentioned at the beginning, explaining the dystrophin protein, many of the mutations are deletions to one or more exons that encode this repeat region in the middle of the protein. And so that shifts the C-terminal coding piece of the gene out of frame. But you actually can remove one or more of those exons and restore the reading frame. And what you end up with is a version of this protein that is internally truncated and is missing some of these spectrin repeats, but is actually still largely functional because if the N-terminus and the C-terminus are intact, it can still serve as that functional shock absorber inside muscle cells. And so our goal was to not necessarily restore the piece of the gene that was missing, but actually restore the reading frame. So what does that actually look like in practice? Well, zooming in on the exon structure of the dystrophin gene, I'm showing this hotspot region of mutations between exons 45 and 55 up here at the top. The arrows indicate how these exons fit together. So dystrophin gene in total has 79 exons. About half of the mutations occur in this region between exons 45 and 55. As I mentioned, it's usually a deletion of one or more exons. And so a common type of Duchenne mutation is shown in this middle schematic, where the patient has inherited a deletion of exons 48 through 50. You see now 47 and 51 don't fit together. And so that means that 51 is out of frame. And the C-terminal piece of the dystrophin protein no longer gets made. You get no dystrophin protein. However, if you remove exon 51, in addition to removing 48 through 50, now you see 47 and 52 do fit together. So by removing 51, you've restored the reading frame of the gene. And now this protein gets made, it's only missing the piece that is encoded by 48 through 51. But as I mentioned, that can still lead to a functional linker or tether in that skeletal muscle tissue. And so that's an approach that we and others have taken for restoring dystrophin by being able to just removing gene sequences rather than having to introduce them. Now, there's a number of other possible ways you can imagine trying to edit this gene, and we've explored those as well in parallel. But the focus has largely been on exon deletion for a number of reasons. One is that by removing the exon and doing so very precisely, we have a good idea of what the protein product is going to be. So some of the other types of strategies, you might get variable different protein products. And we think that being able to know what the protein product that you're generating is going to be important to move this forward. Also, in order to cut out exons, we're actually making guide RNAs that target the Cas9 enzyme to the intronic regions flanking the exons. The exons are small, often just a couple hundred of base pairs. The introns are large, sometimes hundreds of thousands of base pairs. So by targeting the intronic regions, we have an incredible amount of flexibility to test many different guide RNAs, thousands or tens of thousands of guide RNAs, to find the ones that are most specific and most efficient, to find really robust targeting tools. And so, so that's been a nice thing to take advantage of. And then finally, I'm showing you an example here of removing exon 51 to restore the reading frame. And that's applicable to about 13% of the patient population. I'm showing here that removal of 51 can treat a deletion of 48 through 50, but you could spend some time looking at the schematic and realize that, okay, it can treat 48 through 50, but it could also treat 46 through 50 or deletion of 51. There's a couple of other mutations that would fall into the category of things that are correctable by exon 51 removal. However, actually, if you do multi-exon skipping, so for example, I told you half of the mutations occur in this 45 to 55 region. And if you look at the schematic, 44 and 56 fit together really well. And so actually, if you just use a deletion strategy to remove all of 45 through 55, that one composition and one strategy can treat about half of all of the Duchenne patients. And so that's something else that we have also demonstrated. I'm not going to take time to show that here. But I will show you quickly an example of how this works. So in this case, in some of our earliest studies, we got patient muscle cells, myoblasts, from a patient who had one of these delta 48 through 50 inherited deletions. We designed guide RNAs targeting the intronic regions, flanking exon 51. And we show that when we treat with both of the guide RNAs, only when we treat with both guide RNAs and the Cas9 enzyme, we show that we get a genomic deletion of that region. I'm not showing the DNA results here, that's already been published, but we can take those muscle cells, differentiate them in culture into myocytes so they upregulate the dystrophin protein. And we can look at both the DNA, the mRNA, and protein production. And what you see at the mRNA level is that if you treat with no guide RNAs or only one of the two guide RNAs, that only goes to one side of the exon, the RNA that gets produced is the full RNA that's missing 48 through 50. But when we treat with both guide RNAs, now we have a new species of mRNA, and we can actually, that is smaller because it's missing exon 51, and we can go in and sequence that cDNA and confirm it's the exact junction of 47 and 52 that we intended to create. And we can do a Western blot for the dystrophin protein to confirm our hypothesis that that upregulates dystrophin protein production, and that's shown at the bottom here. So this exon deletion strategy can restore this dystrophin protein in patient cells. But from there, we wanted to be able to show that we can do this in vivo. That's the real challenge, right? It's kind of easy now to do genome editing in vitro. It's the in vivo part that can be difficult. And so for that, we had to move to a mouse model of the disease. And in this case, the commonly used mouse model of muscular dystrophy is called the MDX mouse. And this is a mutation to the mouse dystrophin gene. It's actually a spontaneous point mutation. So it's a little bit different than the type of mutation that you would find in a Duchenne patient. It's a premature stop codon caused by a nonsense mutation in exon 23 of the mouse gene. But the idea of exon deletion still applies because it turns out that if you cut out mouse exon 23 in the MDX mouse, exons 22 and 24 fit together perfectly, and you can restore that dystrophin protein that's not previously otherwise made in that mouse. And so we were able to go forward and make guide RNAs targeting the intronic region flanking mouse exon 23. We combined them with a small Cas9 that we can fit into a conventional adeno-associated viral vector, which is a commonly used gene therapy vector. There's actually 2 approved gene therapies using the same AAV that we used here. And we can package that AAV with the Cas9. In this case, it was on 2 different vectors. We've also done a version of this experiment where we put everything in one vector. But we mix the Cas9 expressing vector, the vector encoding the 2 guide RNAs targeting the sites flanking exon 23, and we inject that into the mouse and then analyze the muscle tissue. Now, to give you a sense of what the dystrophin protein distribution looks like in vivo, I'm going to show you a cross-section of normal wild-type muscle. So there's a cross-section of a leg muscle where we're staining all of the muscle fibers for the dystrophin protein. And what you see is all of these fibers are coated with dystrophin, and it's serving as that molecular glue that is holding all of those muscle fibers together. This is a wild type mouse. If you do the same cross-section and staining of an MDX mouse, it looks like this. There's no dystrophin protein present. But in that MDX mouse, where we injected the viral vector encoding the CRISPR-Cas9 system, we waited 8 weeks, and then we stained for dystrophin, it looked like this. And so this was really exciting for us. We can quantify this and see that about almost 70% of the dystrophin of the muscle fibers are now dystrophin positive. We've restored the dystrophin protein. There's a lot of other details about the efficiency of gene editing and how that's different at the DNA and RNA and protein level, but they're all in this paper. And so since it's published, I'm not going to take the time to go through all of that. But the fact that almost 70% of the muscle fibers were now dystrophin positive was sufficient to give us confidence that this could potentially improve the muscle function, that this dystrophic, degenerating, weakened muscle might now be protected and get stronger. And in fact, when we used assays to look at muscle strength, whether it was twitch force, tetanic force, or looking at fatigue over multiple rounds of contraction, in all cases, those muscles got stronger. Now, they didn't exactly get back to wild type muscle. We can't create new muscle, right? The muscle that's lost, weakened, is already gone, but we can protect and restore that muscle compared to untreated, and that's what we're showing here. So this work was now published 5 years ago, and it generated a lot of excitement at the time, and as a new way to apply Duchenne, and also because this was one of the earliest examples of actually taking this new CRISPR technology and delivering it in vivo to a mouse model of a disease with a clinically translatable approach and showing that it could be successful. It also got a lot of attention because there were a bunch of papers that came out at the same time or shortly later showing very similar approaches. So when we published our work, both Amy Wager's lab at Harvard and Eric Olson's lab at UT Southwestern had companion papers next to ours showing similar results. And this is just some of the many other papers that have come out since then, pursuing similar types of strategies. I think what I find exciting about all of this different work is that there's a lot of different things about these studies, right? So it's first of all, it's great when lots of labs can reproduce the same result, right? It means it's pretty robust. But if you look across all of these different studies, they're all applying gene editing to different animal models of Duchenne. But they're using different guide RNAs, different Cas9s, different AAV vectors, different doses. They're treating at different times. They're looking at different outcome measures. Everything about these studies is very different, but that also indicates that, again, it's a robust strategy that's resilient to all of these different changes in the parameters. It probably also means that there's a lot of optimization that can be done to improve this by finding, what the optimal criteria are across all of those different parameters. But one of the things that was interesting to us is that, even though there was a lot of excitement about this approach, it was still very early and there was still a lot of unanswered questions, right? And a lot of things on our mind that we were worried about moving this forward. And I'm going to walk you through some of those now. We can talk about some of the work that we've done to try and understand some of those issues. And so some of these are listed here around immunogenicity, durability, progenitor cell targeting, and then the potential to get a full-length dystrophin protein. And so I'll go through these one at a time. So first, a very common concern in the gene editing field is whether it's CRISPR or any of these other gene editing enzymes, almost all of them are made of components that are foreign to humans, right? CRISPR is a bacteria-derived system. And so the current concern is that if you take this machinery and put it in vivo, there's going to be an immune response against the gene editing machinery and potentially against the gene corrected cells. And so we wanted to kind of understand that and look more carefully at it. And so when we looked at those mice that we had treated with the AAV vectors encoding Cas9, first we looked for an antibody response. And in fact, we did actually see an antibody response against Cas9. And this is true whether we used an AAV8 vector or an AAV9 vector. These are two different serotypes of the virus. Both go to muscle pretty efficiently. It doesn't matter whether we deliver intramuscular or intravenous. In all of these cases, we get a very robust antibody response against Cas9. Interestingly, that only happened when we treated the adults. When we treated neonatal mice, these are mice that are 2 days post-birth, we did not see an immune response. So at least in mice, and that you can kind of get past the immune system by treating early while the immune system is still developing. So this is an antibody response, which actually is not really a concern for us because the Cas9 is supposed to be inside cells. So antibody in the serum is not really going to prohibit our system from working. But what we were worried about was anti-Cas9 T cells. And so if T cells are surveilling for Cas9 expressing cells, that could be potentially a problem. And in fact, we do see T cells reactive against Cas9, again, only in the adult mice, not in the neonatal treated mice when we treat with the AAV vector. Now, a couple of caveats here. I mean, there was no immunosuppression used in these studies, which immunosuppression is now basically a standard regimen that gets introduced with any gene therapy because of immune responses against any component of the gene therapy. We also used a ubiquitous CMV promoter in these studies. And we think that potentially being able to regulate the promoter, use cell type specific promoters might limit this type of immune response. But it's pretty clear across our study and a bunch of other studies in the field now that the immune system is going to find and see this Cas9 protein. But that begs the question of like, well, wait a minute, we saw these kind of nice results where the dystrophin protein was getting made and the muscles were getting stronger. You know, if there's an immune response against the Cas9, you know, what could be going on here? And so that led us to ask the question about the durability. Is it possible that this immune response is getting in the way of a durable effect? And so for that, we actually went past those 8-week experiments that we originally did and looked at longer term results by 2 different approaches, either doing an intramuscular injection in adults and looking at 8 weeks and 6 months, or doing a systemic IV administration in neonates and looking at 8 weeks and 1 year. And what we saw was durable production of the dystrophin protein. So it did decrease some in the adult treated mice. In the neonatal treated mice that got the systemic administration, it either was stable or increased. And this is looking at the DNA editing frequency, but now we're looking at the dystrophin positive fibers. And you can see all the way out to 1 year in the neonatal treated mice, we get nice restoration of that dystrophin protein. So you know, so we do see that the immune system recognizes Cas9, but I still think it's, you know, a little unclear as to what the immune system is doing in response to that observation. And I think in our study and in other studies in the field, there is reports of long-term durability of gene editing and the downstream effects of gene editing. So I think it basically highlights that more work needs to be done to understand what the immune system is doing and potentially be able to control it. Now, the fact that we were getting durable responses, even though we know that, you know, muscle cells turn over and there's always kind of a regeneration process going on in these muscles, led us to ask the question around, you know, we're pretty confident that we're editing the muscle fibers, but what about these muscle progenitor cells? And the muscle progenitor cells in vivo are called also satellite cells. And as I mentioned, the myofibers are these very long cells that span the length of the muscle tissue that have hundreds of nuclei, so multinucleated fibers. The satellite cells are the kind of adult stem cells of muscle that are mononuclear cells that sit just outside the muscle fiber. And in response to a regeneration stimulus or damage to the tissue, those satellite cells will asymmetrically divide. One of those cells just self-renews and becomes a stem cell, but the other differentiates, fuses into the muscle fiber, and contributes new nuclei and participates in muscle regeneration and muscle fiber repair. So the question was, you know, we're pretty confident that we're editing these myonuclei in the myofiber, but what about the satellite cells, and could those be contributing to durable responses? For that, we acquired a mouse where the satellite cells are labeled with green fluorescent protein. So the master regulator of the satellite cell gene expression network is a transcription factor called PAX7. And these mice, one copy of the PAX7 gene has been labeled or targeted with GFP. So the satellite cells are going to be GFP positive. And we can actually take the muscles from those mice, isolate the mononuclear cells, and use flow cytometry to capture the satellite cells. And you see here in this scatter plot that about 5% of the mononuclear cells in a skeletal muscle tissue are PAX7 positive, and those are the satellite cells. So then we can cross this mouse to our MDX mouse, deliver those gene editing reagents that excise exon 23 and study whether or not those GFP positive cells are also getting edited. And in this schematic I'm showing you, in fact, they are. So you see that we can isolate the GFP positive and negative, and we're getting nice excision of exon 23 in vivo following administration of that AAV CRISPR vector, suggesting that, in fact, yes, we are getting satellite cells edited. And this work that we did really followed up on some beautiful work from Amy Wager's lab at Harvard that was the first to show that actually they were also getting gene editing of satellite cells in vivo by this type of approach. Okay, so then the last piece of this that I wanted to share is around the idea of being able to restore a full-length dystrophin protein. Now, I started this whole concept by saying that you can get these internally truncated dystrophin proteins and they'll do the job, right? They'll still serve as that mechanical tether from the cytoskeleton to the outside of the cell. But that's not totally true. We also know that those proteins are not as good as the full length. They can help, but they're not going to be as good as the full length wild type complete dystrophin proteins. And so we did want to continue to think about how we could potentially get to realize the full promise of applying gene editing to a genetic disease, right? If you think about gene editing being able to make any change to the genome sequence at will, what you'd really want to do is be able to take genetic mutations and restore them back to wild type. And remarkably, almost all the clinical trials that are happening right now with gene editing, they're not fixing genes, they're breaking genes. Because as I mentioned at the beginning, it's easier to break genes than it is to fix them. And so I think that the field has some work to do to figure out how do we get to realizing that vision of gene correction. The other piece that we wanted to address at the same time was being able to use compositions that target the human gene in vivo. So one of the challenges for the gene editing field is that because we're treating underlying genetic mutations, we're not treating the symptoms of a disease, or we're not trying to treat compensatory mechanisms, we're actually trying to fix the underlying genetics. Using animal models, like the MDX mouse in this case, only lets you test CRISPR compositions to correct the mouse mutations. I already told you that mouse exon 23 doesn't look like the human gene, and that's not where the human mutations typically occur anyway. And so for that reason, we went ahead and developed our own humanized mouse model of Duchenne. So in this case, we were lucky to be able to acquire from a group in the Netherlands a mouse where they had knocked in, actually they didn't knock in, they just made transgenic via artificial chromosome, the full-length human gene. This is actually quite a feat. The full-length human dystrophin gene is actually the largest gene in the human genome. It's about 3 megabases. But they managed to get this whole thing integrated onto chromosome 5 of the mouse genome and then cross it to the MDX mouse. So now this mouse has the full-length human gene with all the introns, exons, promoter structure, but doesn't make the mouse dystrophin protein. However, the mouse that they had generated was a wild-type normal mouse, and so that doesn't help us for using gene editing to correct mutations. So we actually used germline CRISPR editing in these mice to remove exon 52. That would then create one of these Duchenne-like mutations by pushing 53 and downstream out of frame. We characterized that mouse, and so now you see there's basically with the delta 52 deletion, there's no dystrophin being made. We can look at that by immunofluorescence. We can look at it by Western blot. And we can also characterize the features of these mice and show that it actually leads to a dystrophic phenotype because they're not making mouse or human dystrophin now. And so they move slower and they're weaker. And so then the goal was to try and use these mice to develop an approach that restore the full wild type dystrophin gene. And for that, if we want to be able to correct this exon 52 deletion, we need to find a way to be able to push exon 52 or insert exon 52 back into its appropriate place in that region of the human gene. I already mentioned that homology-directed repair was not going to be a very effective method for using this because it's shut down in these post-mitotic cells. And so the question was, how can we leverage this active pathway, non-homologous end joining, to restore this full-length dystrophin gene? Several groups had already previously published a method to do this by basically delivering a linear piece of double-stranded DNA that then the non-homologous end joining mechanism can grab onto and ligate into the genome. And one group had termed this homology independent targeted integration, or HIDI. And so this was the approach that we wanted to explore for correction of our humanized Duchenne model. Okay, so if we look at the wild type human gene, we've got all the normal exon structure, exons 50, 51, 52, and 53. In our Duchenne model, we've deleted 52. And so the approach we took, and now 53 is out of frame, it doesn't fit with 51. The reason I'll point out why we removed 52 is that it's actually correctable by a number of strategies, including removing 51, removing 53, removing 45 through 55, or inserting back 52. And so the approach to deliver back 52 was to have a guide RNA cut site in this intronic region between 51 and 53, and to deliver these two vectors. The first encodes the small Cas9 enzyme that we can fit on AAV. The second vector has a guide RNA targeted to that site downstream of exon 51, and it also carries a copy of exon 52. And that copy of exon 52 is also designed to have those two guide RNA target sites, or have the same guide RNA target site downstream of exon 51, flanking exon 52 in this donor vector. So if all goes according to plan, then both of these AAV vectors get into a cell. The guide RNA gets made in complexes with Cas9, and then simultaneously cuts at both places around exon 52, liberating it from the AAV vector, and also cuts in the genome. At that point, it can become a substrate for non-homologous end joining, where it grabs those two loose ends in the genome and ligates them into exon 52, leading to this targeted integration of 52 in the genome. You'll note that when the 52 goes in the correct orientation, then the guide RNA target site gets ablated, right? The rectangle and triangle no longer fit together down here in the corrected version. If it goes in backwards, then the guide RNA sites do get reproduced on both of those locations and can be recut and recut and recut until it flips around and goes into the correct direction, at which point it can't be recut. So it's kind of a thermodynamic trap for getting the orientation of integration that we want. So the experiments that we did, starting with, we started with cultured cells from these mice, and we transduced them with the AAV vectors, differentiated them into myofibers, and collected DNA, RNA, and protein for analysis. And what you see is that with two different guide RNAs, guide RNA, we'll call it guide RNA 12 and guide RNA 7, we were able, by analysis of the DNA by PCR, to get that corrected inserted product that we were looking for. And then at the RNA level, we see that we now have mRNA transcripts where 52 has been inserted downstream of 51. And we get protein, full-length, complete dystrophin protein production only when we deliver both the right guide RNA and exon 52. And we tried this at different ratios of Cas9 vector to donor vector, and generally observed that more donor vector is helpful in getting more targeted integration. That was just in vitro results. We then want to move in vivo. So at day 0, we do a local injection into the leg muscle of these adult mice. Again, we tried the two different guide RNAs and two different ratios of Cas9 vector to donor vector. And 4 weeks later, we look for dystrophin expression. Here at the DNA level, we see that we basically get stronger, more consistent results with guide RNA 7, although we also get some signal with guide RNA 12, but we're clearly getting targeted integration to the site in the genome that we're looking for. We can, again, look at the RNA level. We see exon 52 has been inserted in vivo, and in vivo, we're now getting that dystrophin production. You can see with PBS, there's no dystrophin protein being made. And then in all of these various conditions across 3 replicates per condition, we're seeing restored full-length dystrophin protein. Now, the approach that I just described, though, for insertion of exon 52, it's great. I think it's great that I guess that we were able to show this targeted integration to restore a full-length gene. But the approach I just described would only be applicable to a patient who is missing exon 52, right? That's not going to be that many patients, right? A very small subset of Duchenne patients would be missing 52 and only 52. And as I mentioned at the beginning, one of the general challenges for developing gene editing therapies for genetic disease is trying to have a strategy and a composition that can get to a large segment of the patient population. Now, in the case of the Duchenne patient population, I already mentioned that there's actually a hotspot region in 45 through 55 where most of those mutations occur. And so we then hypothesized, well, what if there was one strategy that we could get to all of these patients? And so the idea was, what about instead of inserting exon 52, what if we made one super exon that encodes all of 52 through the rest of the gene, all the way through exon 79, and we insert that downstream of exon 51? Now that approach would be applicable not just to patients who are missing 52, but a patient who's missing anything downstream of exon 51, which actually would get to more than 20% of the patients. And in fact, there's a strategy that we didn't pursue here because we didn't have the right mouse model, but we could fit all of 45 through 79 into one AAV vector, and that would get to about half of the affected Duchenne patients. So now in this case, we changed out our donor vector, so it encodes this one cDNA of all of 52 through 79, and we deliver that with the idea that that'll get inserted downstream of 51. It'll push 53 and whatever else is encoded in that patient's dystrophin gene downstream, away, should not be transcribed, because we have both our stop codons in all three reading frames, as well as a polyadenylation signal. So it should terminate transcription at that site. And again, as I mentioned, we could potentially expand this to an even larger region of the gene, but we focused on 52 through 79 since the Delta 52 mouse model was the mouse we had available to us. Same type of results here. We repeat that study with this new larger super exon donor vector. We show that we can insert that super exon into the genomic DNA. We show that it gets encoded in the mRNA, and we show that with the super exon, we do get full-length restored dystrophin protein. And then we can also test this in vivo with a similar strategy that I already described. And again, it's going to be less efficient because we now got a bigger piece that needs to find its way into that site. And you see at the genomic DNA level, the signal is relatively weak, but we do get nice, robust detection at the mRNA level. And we can see that it's low levels, but we do see some amount of that full-length dystrophin protein being made in those treated muscles. All of that was just treating one leg muscle of an adult mouse. We then wanted to take it one step further and do a systemic administration. Because of the doses of AAV involved and the biodistribution, we went to smaller, again, neonatal mice. We inject those again at day 2 after birth, and wait 8 weeks to analyze the tissues. Again, we can look at DNA, RNA, or cDNA. We can quantify this with droplet PCR and see that we're getting about 20% of the mRNA transcripts are now encoding either exon 52 if we use that donor vector, or the super exon if we're using that. Here's an example where we took a cross-section of the heart tissue of these treated mice and stained for dystrophin protein. And you can see that we're getting, in our most efficient strategies, we're getting about 10 to 25% of those cardiomyocytes to become dystrophin positive, although you also note that there's a lot of variability across these different mice and across different tissue sections, which I think further highlights the need to solve some of the delivery challenges that continue to persist for getting genetic cargo to all of the affected muscle tissue in the body. Here I'm showing the Western blot for the heart. Again, you see there's no dystrophin being made when we use the scrambled guide RNA, but when we use the correct guide RNAs with the different donor vectors, we get full-length dystrophin in the heart. And then as kind of a functional measure of whether we're preventing muscle degeneration, we looked at serum creatine kinase levels. Creatine kinase is an enzyme that should stay inside the muscle tissue. When the muscle tissue is damaged and degenerating, it gets released into the serum. And so you see that in a normal healthy mouse that has the full-length human dystrophin gene, serum CK levels are low. In the control-treated mice with the scrambled guide RNAs, the CK levels are high, and we're able to reduce those serum CK levels by about half with our treatment. And so there's still work to be done to improve efficiency, but this at least demonstrates that we're having a functional impact on the muscle degeneration and dystrophic phenotype with this type of approach. So just to summarize and finish up the talk, a strategy here was kind of building on work that we and others have already done for years on removing exons or disrupting splice sites to exclude exons, but actually trying to get more towards the full promise of gene editing to restore full length wild-type proteins using this HIDI-based approach. And to just summarize the complete talk, I wanted to kind of give you an example of where we've applied gene editing to a particular disease area, as well as some of the general problems, or not problems, but issues that we've encountered and continue to try and overcome around things like immunogenicity, durability, targeting progenitor cells in vivo, and then the types of edits that you can get, whether it's removing something, breaking something, mutating something, exchanging something, or in this case, integrating something. There's a couple other things I didn't talk about, like some of the other unintended genomic outcomes that can happen. If that's of interest, I'm certainly happy to take that on in the Q&A. But I think collectively this highlights, the need to continue to do work in these different areas to improve these gene editing systems. That being said, I think it's also clear that even the systems that we have in hands today are doing some remarkable things in some of the early clinical trials that we've been observing over the last year or two. With that, I of course want to thank all of the people who did the work. Hopefully I highlighted each of the grad students and postdocs along the way with their names and pictures for the different pieces of the work that they were responsible for. And for this particular project, I want to thank the support of Sarepta Therapeutics, as well as some of these other groups. A lot of this work is also part of our efforts in the Somatic Cell Gene Editing Initiative, which is a new NIH consortium that I know Columbia is also involved with as well. And with that, I'll thank you for your time and attention and the opportunity to come speak to you in person. And I'd really be happy to answer questions if there are any.
Questions? Just want to remind you that there is upcoming BME symposium. And then before the BME symposium next Friday is Dr. Duchenne from University of Chicago will present quantitative chemical imaging of organelles. And remember about the annual BME and Engineering in Medicine Symposium. You will find that at the bme.columbia.edu. The annual symposium is scheduled on the February 17th.
All right, so we have a few minutes for questions. I think the way we're going to do this is we're going to alternate between the in-person people and the questions from the internet. Sound good?
Sounds good.
Okay, so the first question came from Robert Roth, and his question was, have you researched prime editing for treatment of Duchenne's muscular dystrophy?
Yeah, so that's can I use this mic? Or
Yeah, of course.
Yeah. So the question was about using prime editing for Duchenne, and I think that I'd say as a broader idea, the gene editing field is moving remarkably fast, right? For those that are paying attention, it's every month there's new editing technologies that are emerging. It makes it really exciting and fun area to work in. It's also very difficult to keep up with it, that technologies keep changing as you keep trying to implement them for different applications. I would say that some of these new technologies like the base editing, prime editing, there's some really interesting new targeted integration and recombination strategies, targeted transposition, which Sam Sternberg here at Columbia is the leader in that area, is really exciting as well. I'd say as a general sense, this broad category of editing strategies, I think it'd be really interesting for certain applications, but maybe not everything is going to be good for all of these. So to answer the specific, and so we've applied some of those to Duchenne, but to give you an example to the specific question about prime editing, I think that at least as of now, prime editing is largely focused on smaller size changes. And so, but I know there's been some more recent results in targeted larger deletions, but this idea of a one-size-fits-all approach for getting to a large patient population, I still think is maybe most uniquely addressable with the nuclease approach that we applied here. But I think that, the field has work to do to try and figure out strategies of how these technologies that are more mutation specific can be developed for patients. Because I think that it's going to be a sad story if we have the ability to use patient-specific editing strategies, or it's been something like a base editing or a prime editing, but not be able to move it forward because it's too unique to that patient, and there's not a motivation that there's a large enough patient population to support its development. And so you'll see that there's a lot of these efforts, kind of bespoke gene therapies or N-of-1 gene therapies being developed, and I think that that's going to be really important for us to figure out in order to maximize the benefit for patients for these new technologies. So yes, we've explored prime editing, but there are some nuances about how broad of a patient population it can be applied to.
Thank you. Questions from the in-person audience? I will bring you the microphone. Yeah. No, there is a need because people online need to hear you.
Thank you for the talk. And actually, it's just a small question. The last approach you mentioned, you kind of remove all the exons from the 52 and replace just with the exons. That's why you can fit in the AAV, right?
We remove all the introns. That's correct. That's right.
Yeah, and then you just put back all the exons, right?
Yep.
So in this case, I don't know a lot about this protein. So you just recover the full length protein, right? But probably we can have different splicing variants. And what about if we have different splicing variants?
Yeah, so this is a good point. So the question relates to the fact that if we insert that big 52 through 79 super exon, any splicing that happens downstream of that, we've now gotten rid of because there's no more intron exon structures. And it's a very good point. And there are other alternative transcripts that get made from downstream promoters, et cetera. I think that short answer is there's work to do to understand whether that's a problem or not. I think our hope is that the restoration of the full length is what we need to address this dystrophic phenotype in skeletal muscle. And if we can do that, we think we can help. Restoring all of it is all those different transcripts. Is maybe beyond the scope of what we can do right now with some of these editing strategies in terms of accomplishing efficiency. But I think that restoring the full length in cardiac and skeletal muscle will be beneficial, even if we don't get those other isoforms in other cell types.
Thank you.
Yeah, I also have a question to that extent. How do you ensure with the targeted deletions that you're not impacting splicing? I guess what design considerations do you make for that? And also kind of piggyback off that idea, with kind of the developments in antisense, do you have, obviously you're a little bit biased, but do you have a preference in terms of an ideal therapeutic, whether CRISPR seems to be the stronger candidate or antisense for these exon skipping techniques?
Yeah, so the first question was around, are we disrupting splicing when we delete exons? And yeah, that's a concern. So I presented it as if we take out 51, then 47 will just go to 52 and it'll be perfectly clean, right? But how do we know that the splicing machinery doesn't get confused and start putting 47 together with 64 or some other exon? And in fact, we do observe that there is some odd splicing effects. So we can characterize that by collecting all of the RNA and doing long read sequencing or transposition and sequencing, unbiased sequencing of those transcripts to find all of the new junctions that we make. And we did publish that in that work a year or two ago that showed that we do see some new weird splicing events. Now, whether they're problematic or not, I don't know. I mean, they might just be a byproduct of this. They may be inert, but there's probably more work that needs to be done to investigate that. Again, maybe it comes back to similar to the other question about splicing. If we're restoring the dystrophin protein and if we're helping alleviate the phenotype, then production of some of these other things that are otherwise biologically inert may not matter. But the good news is that we can characterize that and know what's going on. The second question, remind me.
Antisense.
Right. So I didn't cover this, but in addition to kind of gene editing to remove exons, there are other technologies out there for using RNA approaches to modulate splicing that remove those same exons at the RNA level. And so what are the pros and cons of using an RNA targeted approach versus a DNA gene editing approach? Well, I mean, the obvious advantage of gene editing is that, especially if you're editing the satellite cells, it should be a one and done type approach, right? You treat the cells, take out the exon, and now the dystrophin gets made in those cells forever. With the RNA, you need to constantly be re-administering, and getting all of that RNA to all of the cells for long periods of time while avoiding toxicity could be a potential challenge. And even those RNA technologies that are currently being used in humans, the efficiency with which they're restoring dystrophin is pretty low. And I think any dystrophin, these patients would appreciate, but I think the hope would be that with a one and done, more efficient strategy, where we don't have to target all of the RNA transcripts continuously and trying to do that before splicing happens is a challenge as well. But being able to just fix it at the DNA level might have some advantages. Now, that being said, as I pointed out throughout the talk, gene editing is not easy either, right? So it's great that the patients have some RNA targeting possibilities now while the gene editing technologies continue to develop.
Okay, I think we are out of time, unfortunately, but thank you very much.
Thank you.